All right, why don't we get started? Good afternoon, everybody. Welcome to the HANA seminar series for 2000 fall semester 2020. Moving the seminar series online to accommodate for the global pandemic situation that we're in. So it's great to have everybody here. My name is David Skoll. I'm a professor in the Department of Forestry, and I'll be the nominal host for today's seminar. A few rules of the road that I thought I would offer for everybody. It's best to maintain your own position in mute mode. We can mute it here. I think Renee can mute it since she's the official host. If you would like to see the speaker, I would suggest you use speaker view, but for the most part, Dr. Han will be using the full screen sharing. So you'll see the slides presentations mostly. And what we thought we would do is hold all questions until the very end. And if you have questions for Dr. Han, you can put those down using the chat function of the Zoom, which is found in on the bottom of the ribbon on the bottom of the Zoom window right next to share screen. So today it's my pleasure to introduce our speaker, the first speaker in our series, Dr. Kyung-Hwan Han, who is a professor in the Department of Horticulture and jointly appointed in our Department of Forestry, and we're very glad to have him. He's a specialist in molecular biology and genetics as well, and he focuses his research on wood formation, both primary wood formation and secondary cell wall biosynthesis. His applications, the applications of research are very important for the feedstock development processing for bioenergy and biomaterials, and also for improving crop performance, tree performance, for instance, drought tolerance. So today, Dr. Han is presenting what makes a tree, molecular understanding of tree growth. And with that, no further ado, I'd like to turn the remote console over to Dr. Han and take it away. Thank you very much.
Hey, thank you for the introduction, and thank you for this opportunity. I have two goals in my mind as I begin this presentation. One, I'm glad to introduce and share my research program with faculty and students in the department. And secondly, I would like to use this opportunity to trigger a productive conversation and collaborations. I will welcome any collaboration, any type of collaboration. That would be great. So let me begin. Oops, how do I? Okay, so the outline, I would like to go over on so the secondary growth, i.e. wood formation, and then move on to secondary cell wall biosynthesis, and then I would like to wrap up with this presentation with the biotechnological implications of the findings. So although my main interest is in understanding how tree grows at a molecular and biochemical physiological level, but I'm also interested in biotechnological application of those findings. So when I was student in the department, I was student between 1986 and 91. Jim Hanover, this seminar series actually named after him. Jim Hanover was on my committee, and he, at one occasion, he pulled me aside and told me that if I don't have any applications, my fundamental knowledge does not mean much, does not carry any further. So I thought that was good advice. And since then, I've been trying to understand how tree grow and then trying to apply those knowledges, right? So the unique features, as I begin this, my research program at MSU in 1999, I set an overarching question of what makes a tree a tree. So under that overarching question, so first thing I did is what are the unique features of tree growth compared to other herbaceous plants. So the secondary growth and wood formation is very unique to tree species, although a handful of herbaceous plants also undergo secondary growth, but to the extent of wood formation is only unique in tree species. Another unique aspect of tree growth is the perennial growth habits. Like in a temperate region, we do have a summer active growth and winter dormancy. And this active growth and dormancy cycle is very unique in tree species also. And another one is a formation of extractive in wood. That's basis for many industrial applications. So my laboratory has worked on all three aspects. So this one, you're looking at the tree trunk cross-section of black locust, Robinia pseudoacacia, that I collected from Tree Research Center and viewed under UV light. The bright side in here, hardwood, is brighter because of a lot of phenolics. So it does have a lot of chemicals accumulated here. And my laboratory looked at what are the biochemical and genetic changes in this sapwood and hardwood transition zones. And then we worked on annual growth and dormancy cycle. Of course, the secondary growth is the most interesting part of my research program. So what are secondary growth? Typically in herbaceous plant, the plant stems are made of three components: dermal tissue and ground tissue and vasculatures. And in primary growth, these are all derived from shoot apex, apical meristem at the tip of the shoots. That's once they are differentiated into unique cell types, that's the fate of their cell type, and that's done. However, in tree species, there's an additional layer of meristem in addition to shoot apex. There's called vascular cambiums. So these vascular cambium cells are meristemic cells, so they can differentiate into, if they divide it into inside of the stem, it's becoming xylem, which is wood, and then outside the phloem. So it has that potential. So it goes through three-directional growth. And secondary growth, maybe you can simply state that as a diameter growth in tree growth. That's pretty much what it is. So the genetics of secondary growth, when I had that question, overarching question in front of me, so there are three competing hypotheses to explain the unique wood formation in trees. Is this because of the tree species had unique set of genes for wood formation, or tree species acquired new function out of old genes, or tree species had differential expression of the existing repertoire? So as you can see in here, so I'm going to say the conclusion first. So according to my study, wood formation is because of differential expression of existing genes. This illustrates that. So one Arabidopsis herbaceous annual plants, this has only primary growth and procambium here. But when in my laboratory, we developed a system where we can induce this stem to undergo secondary growth, fully developed vascular cambiums in here. And then if you compare these two electron micrograph, this is a stem and wood tissues in Arabidopsis plant. This is popular. Anatomically almost identical, except a few more phloem fibers in popular. Other than that, pretty much identical. So that supports this conclusion. So if you look at the literature, there are a number of observations that can lead to, that can guide you, and actually guided us in our approach. So the early observation was in 1938. The cambial activity is reduced by flowering. Building on this early observation, my colleague and friend Simcha Leviadon, he's professor in University of Haifa in Israel. So he took this Arabidopsis plant out of just curiosity. He took this plant, placed on his windowsill in his house, and when the inflorescence stem comes out, he just cut it off. And when he repeatedly cut it off, the vegetative growth continued. And when he looked at actually underneath the hypochondral region, he saw well-developed wood tissue there, secondary xylem there. Actually, when I was in, so I invited him, actually he visited MSU to discuss this. And then, so at this time, we thought this can be a good system for us to study, to look at the genes that are differentially expressed between these two different plants. But the only one problem is by cutting this inflorescence stem repeatedly, that introduces wounding. So in our gene expression analysis, the wounding effect can compound our interpretation. So what we did, we developed a very novel system. So by growing this Arabidopsis is a facultative long day plant. If you grow them under long day condition, it'll grow and flower and die. However, if you keep them under short day condition, it will not flower, and we were able to keep it until up to 8 weeks without flowering, and it kept growing like this. So we decided to use this system for our basis for the our gene expression studies, right? Additional observation lead to also the supporting our approach. So external pressure may be involved in the maintenance of meristemic status of cambial cells in woody stems. That's early in 1962. And in 1981, application of mechanical force, like weight application on de-differentiated callus tissue, trigger vascular formation even in the callus cultures, in tissue culture. And also, another interesting observation, wood formation is positively correlated with the weight of the crown in trees. So that gives, however, how that's happening is still at the time unclear when I began this studies. So going back to first question, what triggers wood formation? In other words, what is the environmental signal that the plant stem to initiate their diameter growth? What are the triggers for those? So in general, forest genetics is very difficult to practice, actually, because of the long generation time. Even like popular early flowering species take 10 years to flower. So unless you have your grandchildren committed to do the same thing in your research program, it's very difficult to make observation in multi-generations in popular or tree species in general. So given that limitation, I thought we will adapt this Arabidopsis model system. So again, we start growing Arabidopsis under short day condition, like 55 days or 56 days. And then we will move them out to long day condition, in this case, 3 days exposure, and then moving back to a short day condition. And then this case, 5 days long day exposure, and then moving back. So we did that differential exposure time and moving back. When we did that, we will actually had these are all synchronized plants, the same age, same growing, same physiological, pretty much similar, except this inflorescence stem. The short exposure will have very short stem, like 5 centimeter tall. And then as you increase the exposure to long day, you will have fully developed stems in here. So if you look at the bottom of the stem, you can see this secondary growth fully happening in the mature plant. I call them mature, but they are all same age. And then the shortest one does not have any secondary growth. So that's basically the system we use it. And then from here we know the differential height. So secondary growth in here at the bottom of the stem is positively correlated with the stem height. So in here, from here, I hypothesize that maybe the body weight carried by the stem is the environmental cue for secondary growth. I mean, it doesn't make sense and consistent with the early observations that I showed you. So, so the plant body weight induced secondary growth. So we had a ton of data. Due to the time limitation, I can't show you, but we had the conclusion that plant body weight carried by the stem is the environmental cue for the secondary growth, wood formation. Then the next logical question is, then how do the cells at the bottom of the stem know there is a growing body of the stem so that they need to have diameter growth? So that was the question. And if you look at in general plant physiology and biology literatures and textbook, the plant growth hormone called auxin is produced on the shoot apex at the top of shoot and basically transported toward down. So we hypothesize, because of the nature of that growth hormone movement, we hypothesize that maybe the auxin is the signaling molecules. So in order to ascertain the prior conclusion, body weight is the one environmental cue for this. So we had this short, like 5 centimeter tall, short stem. And then we applied a weight in here, 1.4 gram and 2.8 gram of weight, by way of wrapping up this stem with aluminum foils, weighted at this much. And then you can see the weight application had fully developed secondary xylem. No weight application doesn't. So in order to test the auxin hypothesis, what we did is we had this plant. Instead of applying the aluminum foil weight, we decided to use this agar gel, similar to agar block testing in traditional or old-fashioned physiology test. So we had this agar block with and without auxins, and the weight application with agar block without auxin does not trigger secondary growth. However, if you add auxin to the agar medium, then it does trigger secondary growth. So we then take a stem and then put a radioisotope auxin in here, weight application. Indeed, weight application accelerate auxin movement. So that's the that's the system we had. And then we looked at what are the genes differentially expressed at the time of wood formations. So we did a whole bunch of transcriptome analysis, and we came out with this very unique pattern of genes, differentially expressed genes. Most of them were like signaling related genes or transcriptional regulation related genes, things like those, and indicating indeed that there is developmental program triggered by weight and auction applications. So to summarize on the secondary growth part, so major differences in transcriptome. The transcriptome means a set of expressed genes between trees and herbaceous plants are typically in transcriptional regulation, meaning gene expression regulation and unknown genes. And the plant body weight carried by the stem triggers wood formation using auxin as signaling molecules. And the genes that are upregulated during primary to secondary growth encoding transcription regulation and signal transductions. And also, at the time was surprisingly, but not surprisingly, the majority of those differentially upregulated genes are encoding or related to secondary cell wall biosynthesis. So secondary cell wall is actually the vast majority of wood tissues. So I say this secondary cell wall biosynthesis is probably one of the most important biological process on Earth, because biologically, scientifically, secondary cell walls represent a defining feature of xylem fibers and vessels. Without secondary cell wall, vessel will collapse under the negative pressure transpirational pool, and that plant cannot survive. And xylem fibers also, without secondary cell wall, cannot support growing body of tree trunk. And economically, it also represents the majority of plant biomass that we have many industrial use for. So if you picture how this secondary cell wall formation is happening, the major component of secondary cell wall is cellulose and lignin and hemicellulose. Roughly, cellulose is about 50% of secondary cell wall. Lignin and hemicellulose about 25% each, depending on the species and tissue types. There are variations, but pretty much that's ballpark figures. So the way in this diagram shows is the cellulose will accumulate and in the form of cellulose microfibril. It's bundle of cellulose. And then this bundle of cellulose microfibrils will be connected by hemicellulose, and then in between lignin will fill in. As an early portion of secondary cell wall dehydrate, they condense, form a dense secondary cell wall. It's very equivalent of this concrete, I'm showing here, pillars or concrete works. So they have, in the pillar, they have a major iron reinforcement, and then these irons are connected by small wires and then filled with cement. Right? Cement will be lignin, equivalent of lignin, small wires, hemicellulose, major cellulose microfibrils. So that's how it works. So in the beginning, to begin our thinking here to guide our experiment, we did extensive literature searches and pulled down all relevant transcription factor genes and lay down in a very loosely connected model. And this, I don't even call it model, but we just pull together and lay down and then staring at this. And then luckily, so we were able to show NAC12 is a master switch for secondary cell wall biosynthesis. So that this picture represents our system that we were using, and at the time this story was featured in the cover of this plant journal. So that was in 2007, and then at the time we show MIP46 is a direct target of NAC12. Because NAC12 is a master switch, we thought MIP46 can also serve as master switch because its location is higher up, relatively. So in 2 years later, we were able to show MIP46 is actually indeed functioning as master switch for secondary cell wall biosynthesis. And we identified a bunch of target genes. So this is just to illustrate, actually to confirm MIP46 indeed master switch. So when we ectopically upregulate MIP46 in a plant, transgenic plant, so the mesophyll cells and parenchyma cells and epidermal cells do not have secondary cell wall formation naturally. However, when we ectopically upregulate MIP46 in this transgenic plant, we did observe secondary cell wall formation, and we confirmed by staining for lignin and other anatomical observations. And then this one shows in Arabidopsis plant, there's another functional homologue of MIP46 called is a MIP83. When we knock out both of them, plant will germinate, seed will germinate, but become lethal because no secondary cell wall formation, so it cannot survive. So that confirms MIP46 is actually indeed the master switch. Then we develop a sort of inducible expression system. So a chemical called DEX chemical is a glucocorticoid. We construct the gene expression vector in a way that if you spray DEX chemical, the gene will be activated or expressed. If you don't spray, you don't have expression or activation. As you can see here, when we created a transgenic plant, no spray and spray, you can see that's have actually induced secondary cell wall biosynthesis effectively induced by DEX chemical. So we have this system. Then we went back to gene expression analysis again. So with the spray and no spray of DEX, what kind of genes are differentially expressed by MIP46? So we were able to identify many genes, mostly in this cellulose and hemicellulose lignin biosynthesis pathways. So we were able to. So if you look at, as I stated earlier, if you look at the plant biomass, secondary cell walls are mostly three components: cellulose, lignin, and hemicellulose. And cellulose probably the most abundant natural polymer on Earth. So if you look at it in here, beautiful polymer. If you get rid of all the lignin, then you have this very white cotton-like fibers, cellulose. Right? So in Arabidopsis, in order for the cellulose to be made, you need to have three genes, CESA4, 7, 8. And we show MIP46 directly activate all those three cellulose synthase. So we developed, I wouldn't go into detail because it's boring to most of you. So we developed a very temporally expression system where you can have this MIP46 as an effector and then assay the target genes activities based on, this is protoplast. You put these two constructs in here in the protoplast, you assay these, and we also showed biochemically MIP46 protein is binding to the promoters of its target genes. So that's assertion of the relationship. So we now know MIP46 directly target all three CESA4, 7, 8 genes. The next question is MIP46 required for proper function of those cellular synthase? Or are there additional or other alternative regulators? So to address that question, we used all three mutant lines. These are mutant that cannot function. So because of that, you have this plant falling over because it doesn't have strength to support. And using those mutant line, we did a called genetic complementation experiment. Basically, we put new not mutated regular gene to complement what's missing there. And in that approach, we use the promoter that can recognize MIP46 or cannot recognize MIP46. Using those approach, we were able to show MIP46 is actually indeed required, or those three cellular synthase. So that's cellular synthase. And then another component, the hemicellulose, we also showed MIP46 is direct regulator. When we upregulate MIP46, we did see upregulation of hemicellulose biosynthesis, carbohydrate monoclonal antibodies, showing the amount increased. And then it also showed the lignin is directly targeted by MIP46 and upregulated. As I said earlier, epidermal cells do not develop lignin or secondary cell walls normally, but with the ectopic overexpression MIP46, we do see lignin deposition there. So that shows lignin is directly regulated by MIP46. So this model that we had, the latest model we have now, and also this was featured in the Annals of Botany cover pages with this beautiful picture in here, the electron microwave. So MIP46 in here, I told you MIP83 is a functional homolog. So the MIP46 directly regulate secondary cell wall biosynthesis. So now I would like to talk about the biotechnological implication of it. So basically this is so what question. So we learned those things, so what, right? So we developed a new technology called elite tree technology. Basically this advanced biomass tree crop technology is featuring greater wood density, in other words, greater biomass accumulation with accelerated stem growth. So that's the underlying hypothesis here. By stacking two traits like increased secondary wall biosynthesis and accelerated stem growth, we can develop a novel feedstock. So that's what it is. And its diagram shows this technology has three components. One is a MIP46 overexpression that will increase biomass accumulation by way of thickening secondary cell wall biosynthesis. And then J20 oxidase will increase stem height growth. And then DX15 is a utility promoter technology that can drive a gene expression in developing wood tissue specifically. So these three components make up this technology. As you can see, height growth is accelerating and then accumulating more biomass. So this is developing wood tissue specific gene expression, illustrating the efficacy of DX15 utility promoter. So this is based on multiple US and international patents. So first component, let me just illustrate one each of the three components. For component one is MIP46. MIP46 is a gatekeeper of secondary cell wall. As you can see, if you upregulate MIP46, you will get thickening of secondary cell walls. As you can see, this is again carbohydrate monoclonal antibody staining, showing that MIP46 upregulation increase cellulose accumulation. and then same lignin increase and mannose again. So all three components, cellulose, lignin, hemicellulose, are upregulated by MIP46. So J20 oxidase is a plant growth regulator. If you upregulate this, or if you increase sensitivity to it, then you will have accelerated shoot growth. So J20 oxidase is one of the key enzymes in converting GA precursor into active form of GA. And so we decided to use this gene to accelerate height growth and stem growth. As you can see, by upregulating J20 oxidase compared to control plant, stem diameter increase and height growth increase, and other chemical composition also increase. Right? And then third component is developing Xylem, developing wood specific gene expression. So this is epidermal in here, and then wood in here. For developing phloem, cambium here, I don't know, you can see, but the Xylem wood tissue in here. So our promoter will be active in here. So any genes you put in with our promoter will be actively expressed in here. This is an ideal place to express your gene, because wood is a large sink, and if you increase activity, secondary cell wall here, you will increase sink demand. And so therefore photosynthetic activity in the source tissue in the crown level will be increased. And then in the literature, there are several GA receptors in this region, so that makes our approach possible. So we did that, and then so this utility promoter is also patented in the US and I believe in Canada too. So this utility promoter, why do we upregulate in developing wood, the cellulose biosynthesis, lignin, and hemicellulose? Because if you increase biomass accumulation in those tissue, will increase sink strength, and therefore allocation of photosynthesis to sink increased. That will trigger increased photosynthetic activity in the source tissue in the crown level. So that's the idea, whole idea. So under normal growth condition, if you upregulate MIP46, you will have increased secondary cell wall biosynthesis, regardless of conditions, tissues. But under stress condition, like a salt or abiotic stress condition, a group of researchers published in Nature in 2015, they observed a very peculiar phenomenon. So if you expose your plant under salt stress condition, that will trigger upregulation of MIP46 gene expression. However, they did not observe any of those MIP46 target gene expression is changing at all. So that got me thinking, what is going on in here? So that, because of that, I thought my hypothesis is MIP46 expression is up under salt stress condition, therefore protein is upregulated, so higher level of MIP46 protein. However, because of the salt stress or other abiotic stress, maybe this MIP46 protein is subject to post-translational modification or protein degradation. That was the working hypothesis to explain this observation. So to do that, we looked at protein structure of MIP46. MIP46 protein has one MAP kinase docking domain and two phosphorylation target site. MAP kinase is a well-known signaling cascade in eukaryotic cells. It's very highly conserved system. So MAP stands for mitogen activated protein kinase. So this pathway is very integral in stress signaling in both eukaryote and the animal and plant cells. So MPK6 is activated abiotic stress. So we hypothesized maybe MIP46 is targeted by MPK6 when it's activated by salt stress. So that's what we proved in here. I wouldn't go into detail of those. Basically, MIP46, MPK6 mediated the phosphorylation of MIP46 lose functions because of lose its stability. Protein gets degraded by this phosphorylation. We had all genetic and biochemical analysis to show that that's the case. So however, how do we then take advantage of this finding? So what we did, so I told you there's 2 phosphorylation target sites. So we made a point mutation of that site, amino acids. Basically, we changed just a single amino acid or those 2 amino acids in a different mutated forms. We substitute those target amino acid with a similar charged amino acid, but not phosphorylation target amino acid. And when we did that, this MIP46 protein was not targeted by MPK phosphorylation. Therefore, we were able to keep the MIP46 function under abiotic stress condition. So MSU filed a patent on this particular aspect. So we're currently using this system now. So finding that MPK6 is negatively regulating MIP46 triggered us to this pathway, looking at the secondary cell wall biosynthesis or wood formation or tree growth under various environmental stress conditions or under changing environmental conditions. So we are looking at many different environmental signaling pathway to connect that linkings. So another example of utilizing this biotechnological approach to improve planting stock. So if you downregulate one of the key enzyme biosynthetic 4CL, you can reduce significantly the lignin content, which is very value for pulp and paper industry or bioenergy industries. And also, if you upregulate isofoluric 5-hydroxylase gene activity, then you will change SG ratio, meaning there are 3 monolignar components in plants. So one is syringil lignin and guaiacyl lignin and H lignin. So these are making up the lignin polymers. And the higher syringil over G lignin ratio means the lignin can be easily extracted. So that has a value to those industry also. Another approach that we are taking right now, we recently published this approach, the proof of concept actually. So the MIP46 overexpression with a concomitant suppression of lignin. So that the idea is we will reduce lignin but increase cellulose biosynthesis. Therefore, the plant biomass we're harvesting from, say, poplar will be a higher value for pulp and paper and bioenergy industries. So we actually show that in transgenic poplar by overexpressing J20 oxidase and MIP221. This is a negative regulator of lignin biosynthesis. By simultaneously upregulating these 2, we were able to achieve lignin reduction and accelerated growth. That's another proof of concept that we're talking. So we decided to test this in our field. So we generated these electric poplars, and in a greenhouse or lead house, we saw that differences in terms of growth. As you can see, all the star marks are statistically significant compared to control. This is a popular NM6 that we use. It's been known for its performance in Midwestern region, so it's very economic clone. So we use that, and then this is my colleague, a former postdoc, Jae-Hyun did in Korea for 3 years in field testing. As you can see, actually our electrically popular grow fast. So the goal maybe is short rotation intensive culture capable of sustained production of over 22 tons per hectare per year with improved biomass quality. That would be super. This is a popular plantation in Patlatch in Oregon, and their harvesting operation. It's quite amazing. So all the standing tree, from cutting standing tree to the pulp mill within 24 hours. That's fascinating operation there. And then another application, we are actually collaborating with a colleague in University of Sao Paulo in Brazil. This is a teak plantation I toured twice. So teak, in this plantation, they grow about 18 years and harvest it. So if we're interested in testing whether our electric technology will reduce this teak rotation to 15 years from 18 years without compromising the quality of wood. So that we are testing right now. And we do have maybe third year transgenic popular field testing and the Sand Hill plot of the Tree Research Center. So it's ongoing. So we do not have data to present yet, but it's ongoing. Any suggestions or collaboration discussions with regard to the technology I presented or the research I conducted will be welcomed. So to acknowledge, so my former postdoc, and he was also research assistant professor in the Department of Forestry while I was with him. So he and another former postdoc, they moved on to their own independent laboratory in Korea. So they are the ones mostly discovered the research part. And Dr. Jong-hee Im is current postdoc. He also worked with me in the past briefly. So he's also the one post-translational regulation of MIP46 study, environmental and biomass biosynthesis interlocking system. He's studying those. And then Keith Lee, he and I have been collaborating since 1999. He recently retired. So, and Paul, Bridgie, and Randy, they are very instrumental and critical help with regard to field testing, setup, and maintaining. So with that, I would like to thanks for your attention. If you have any question, I will be happy to try to answer. Oops, not this one. All right.
Okay, thank you, Dr. Han. So while we're waiting, we can take some questions if you want to post your questions in the chat area. Then Dr. Han can bring that up and look at that and answer your questions. Okay. Well, that was good. Dr. Han, when I have a question for you, I won't put it in the chat. I'll just use my prerogative as the host to ask it out loud. You talked about the sink.
Right.
You referred to the sink, but in my business, that means one thing. I'm wondering if it means the same thing in your line of work too. Do you mean a sink for carbon?
Right.
Carbon dioxide?
Exactly.
So increasing the biomass.
Right.
this approach will stimulate the increase of biomass over, say,
Right.
Leaf material or fruiting bodies or something like that.
Right.
So do you have any sense of how much that can be with the in a kind of situation like with elite tree systems?
So in the greenhouse data that we have right now, so elite tree popular we estimate about 20 to 30% increase in biomass accumulation.
Mm-hmm.
But that has to be tested in the field condition, because actual growing condition is not, like a greenhouse condition. So that's why we are doing this field testing. But with regard to sink strength, I had that in the slide, but I left at the last minute. So if you look at the sink force relationship within the plant, within the tree, so the wood tissue and vascular cambium sits in the middle. So fruit is the highest. I mean, the seed is the highest demand for highest sink. And then actively growing tissue, and then root and others are lower than the wood tissue. So it actually, if you look at the hierarchy of that sink strength, the wood tissue is sitting in terms of strength, is sitting in the middle, but in terms of volume, it's the highest. So I consider wood tissue is the highest sink tissue. And if you accumulate 20% more biomass in the wood tissue, that means drawing a lot of photosynthate into those, and that will trigger naturally photosynthetic activity. So two things will happen physiologically. If you increase sink tissue strength, either that will trigger upregulation of photosynthesis, or photosynthesis will be limited by the extra demand. So, but that has to be tested by physiologists.
Mm-hmm. I see. Okay, thank you. So I think I'll turn it over, Dr. Han, because there's some questions accumulating in the chat panel.
Right.
That you can take over.
Okay. So I have a question from Akiro. I'm fascinated by the popular plantation in Oregon. Is water a major limiting factor for popular growth? Can you mitigate the growth limitation by gene manipulation and sex selection? Excellent question. Actually, so that the Oregon plantation was in Boardman. Boardman is the east side of the Cascade. Basically, it's a desert area. So they do, so at the time, no longer U.S. paper companies having those anymore, but at the time they had this plantation with the drip irrigation. So they provide the drip irrigation with the fertilizer and pesticide and all those manipulations. So the next question, second question, I think it's a very excellent one. Can we mitigate the growth limitation by gene manipulation? And actually, we do have a technology called Jericho, drought tolerance technology. Jericho is a genes that regulate the ABA homeostasis in the cell. Actually, MSU has 2 patents on it. So if you upregulate Jericho, it will keep the stomata closed, so the transpiration of water loss will be minimized. That's only happening during drought condition. So our second patent is making that Jericho expression only under drought conditions. Regular growth condition, that gene will not be upregulated. Therefore, stomata opens, gas exchange happens, normal growth. So that we do have that technology. And also there are other drought tolerance technologies out there. So yes, indeed, it's possible to mitigate those growth limitations by carefully manipulating those drought period. So, okay, so second question, wow, Lila. she's a friend of mine from Turkey, and wow, she visited us there. So she said, hello, Han. I missed most of your presentation because I did not hear about the talk. May I have the present? Oh, okay, I'll send you, yes, Lila.
Okay.
All right.
Okay, very good questions. Any more for anybody? From anybody, I should say. Well, this is a great talk, Dr. Han, from the smallest molecular level, at least from my interest, from your smallest molecular level all the way up to the global carbon cycle, about what it makes a tree a tree. Very good. So I think if there are no more questions, we are getting close to the planned conclusion time at 4:30 for the seminar. I guess I can't invite you all for refreshments together, but you can certainly have your own refreshments at your own leisure in your own place following this. So I will thank you, Dr. Han, for the great kickoff for the seminar series. Hanover seminar series is greatly it was quite appropriate that you worked with James Hanover yourself, so that's a great tribute to the seminar. And I want to remind everybody that the next seminar is September 22nd, which is a week from now, and we'll continue the series from there.